One critical thing you must understand about combinatorial libraries is the means of calculating diversity. For this, let's consider combinations of gumballs. Suppose we have a gumball machine and there are 20 different colors of gumballs inside, each occurring at the same frequency. If you pick 6 of them and get sequences of 6 colors, how many different permutations of these sequences are there? So there are 20 at the first position, 20 at the next, and so on for 6 positions. So there are 20 to the 6 permutations, or around 60 million variants that potentially could exist within this space. When transforming plasmids into cells, it is generally assumed that by far the limiting step is transformation. You can assume that there are a few orders of magnitude more DNAs in the reaction than actually get into a cell, so you only need to consider transformation as the limiting step. This of course assumes that you are using cells in your experiment. For all in vitro experiments, you must use other strategies to determine the diversity, which involves following how much DNA is present at every step of the experiment. You should assume that each colony is derived from a single unique member of the library. So the diversity of a library is the number of colonies observed following transformation. It is very common for multiple plasmids to enter a single cell during the concentrated conditions used in these fabrication schemes. You must use a lot of DNA to hit these high colony counts. However, that extra diversity is difficult to measure and will depend on the quality and amount of your ligation product. For large libraries, the existence of this co-transformation background can affect the outcome of selections, but does not erode the confidence that your library is sufficiently inclusive. Your diversity is minimally the number of transformants you observe. It might be a little higher in practice. Due to the large numbers involved, this is usually quantified by plating dilutions of the library on multiple plates and then back calculating the diversity. This is referred to as titering the library. When you construct a combinatorial library, you are not explicitly synthesizing each member of the library. You are sampling this combinatorial space randomly, so there are issues of diversity inclusion in your library. So in practice, you need to make more library members than are actually present in the theoretical library. Let's say I glue together 6 gumballs together into hexamers. The analogy of the gumballs is to amino acids, hence 20, or to bases, of which there are 4, or to a set of parts that could be any number. Each member of the library can be considered as the list of chosen members at each site, and here we represent that as a hexamer of gumballs glued together. How many of these random hexamers do I have to make to have a 99% probability that I have at least one of each theoretical member? N here is the theoretical diversity, which we calculated already as 60 million. P is the actual diversity that we'll measure when we titer the library. So we need to determine what value of P is sufficient to have confidence that at least one of every theoretical member is represented by the library. That calculation, whose derivation is provided in the reference bottom right, is given by P equals 1 minus e to the negative P over n. If we set that value to 0.99, we can solve for P in terms of n. And we find out that this all boils down to a simple calculation of multiplying the theoretical diversity by 4. The other issues with sampling are bias and general library quality. Either of these mechanisms can lead to a library that does not contain all theoretical members. When you order ends in an oligo sequence, you don't always get an equimolar mix of bases, so you need to sequence a few clones and confirm that they came out well balanced. Over the years, oligo quality has improved, and usually this isn't a big problem, but you do need to check it. If they are not random, resynthesizing the oligo usually will fix the problem. Even if the oligos are truly random, some methods used for fabrication, notably quick change, can cause bias due to the biased annealing of the oligos. Let's say you put an extended run of ends in an oligo. When that sits down on a DNA during PCR, those oligos that have higher homology to the substrate will have a higher TM and under some circumstances will preferentially bind. This can be improved by making sure you include at least 20 base pairs of homology in the annealing region of your oligos and avoiding any situation in which you are annealing to end-containing regions of an oligo against each other. And that's the problem with quick change. You're putting ends right against each other with the two oligo sites. In the early days of doing this, much interest was in antibodies, and many of these genes were toxic to the cells. When you make libraries, many of the variants will be altered not just in their molecular function properties, but also in terms of their expression and toxicity profiles within the cell. So some members will grow better than others and can, in the worst case scenario, take over the library. You can also cause bias by growing your cells unevenly or over-diluting them during an outgrowth step. For example, let's say you spread your library on plates, but you do not do so uniformly. You get some regions of the plate that are very sparse and others very dense with colonies. Since colony size is dependent on the local density, you get smaller colonies in the dense regions. As a result, those colonies that landed in a sparse region of the plate will become overrepresented in your library, causing bias. There are also some general quality issues you must consider in your library. For example, if you do not be careful to choose a vector in which the insert is 1/4 to 1/3 the size of the entire plasmid, you'll get monocut vector background in your digest that gives rise to a parent vector background in your library. So it is not uncommon to make a large library that in practice is 90% parent plasmid. If you get this problem, you can fix it by pasting in a different size spacer into your plasmid and then making a new digest from that clone. The processes of oligo fabrication and assembly can cause other kinds of errors. It is not uncommon to have a high frequency of point deletions around NNK sites in a library. This often can be resolved by resynthesizing the oligos, but sometimes it is caused by the assembly steps, in which case you might consider a different fabrication strategy. If you do NNK mutagenesis, there will be TAG codons present. If you are seeing TAAs or TGAs, then those aren't supposed to be present and you should be worried. If you are seeing TAG codons at a rate higher than 1 in 32, then there is a bias in your library that is likely resulting from toxicity of the full-length protein. Several gene synthesis companies offer combinatorial libraries as a product. Personally, I've never used such services since they tend to be expensive. However, combinatorial library fabrication can be very difficult, particularly for someone doing it the first time in a setting devoid of experience. So it's something to consider. Whoever makes the library, there are a finite number of methods used to make them, so let's go through them.